Hello and welcome to Teach Me in 10. In this episode, we're joined by Dr. Shaveta Goyal, Group Leader of NGS at Takara Bio USA. Shaveta will guide us through the key considerations for choosing the right RNA-Seq workflow for your research, from deciding when to use ribodepletion or unique molecular identifiers to understanding the importance of stranded libraries. She'll explain how these choices directly impact data quality and biological insight.
Hi, my name is Shaveta Goyal and I am R&D Group Leader in NGS team at Takara Bio USA. Today, I will be talking about total RNA sequencing, and title of my talk is Choosing the Right RNA Sequencing Method, What Really Matters. Rooted in the central dogma of biology, DNA to RNA to protein, RNA sequencing unlocks the dynamic layer of gene expression by revealing how genetic information is actively transcribed and regulated. RNA sequencing has become the gold standard for transcriptome profiling. It enables unbiased profiling. Unlike targeted approaches, RNA sequencing does not require prior knowledge of genes, allowing us to detect novel transcripts, gene fusion, and alternative splice variants that might otherwise be missed. RNA sequencing provides a comprehensive view of cellular function, enabling researchers to quantify gene expression levels accurately across a wide dynamic range from single cell to bulk tissue. Together, these capabilities allow RNA sequencing to deliver a complete and detailed picture of gene activity within cells. This power enables precise insights into cellular states, disease mechanisms, and biological function that cannot be captured from DNA alone. For the dynamic RNA, there are various technologies available for analysis, of which mRNA sequencing focuses only on poly A tail coding transcripts, which represent a small fraction of total RNA. It's widely used for gene expression analysis, but it misses non-coding RNAs. Small RNA sequencing targets short regulatory RNAs such as microRNA and small interfering RNAs. These molecules are low in abundance but important for gene regulation and biomarker discovery. Total RNA sequencing, shown here, offers the most comprehensive view. By depleting ribosomal RNA. It captures all RNA species, including mRNA, long non-coding RNA, and other non-coding RNAs. This makes total RNA sequencing especially powerful for studying alternative splicing, novel transcripts, and non-coding RNA function, providing a more complete picture of cellular activity. One thing worth noticing here is that 80 to 90% of total RNA is rRNA. So to capture what really matters, it is important to remove the abundant ribosomal RNA. Like other NGS assays, RNA sequencing also has three key steps: library preparation, sequencing, and data analysis. Library preparation has few major steps. The success of each library preparation step affects the quality of data generated downstream. Today, we will go through some of the most important factors to consider when generating your RNA sequencing library. One of the things to consider is strand specificity. Strand specificity adds valuable information to RNA sequencing. Compared to standard non-stranded workflows, stranded library preparation preserves the direction of original transcripts. This allows us to determine which DNA strand was transcribed, distinguishes overlapping genes on opposite strands, and accurately detect and quantify antisense regulatory RNAs. Overall, strand-specific data leads to more precise interpretation of gene expression. If an RNA sequencing library is not stranded, it becomes impossible to determine the original transcriptional direction, leading to ambiguity in gene expression analysis, especially for overlapping genes and antisense transcripts. Another thing to consider is amplification bias, which is a major source of error in sequencing data. During PCR, the same RNA fragments can be copied over and over again, creating duplicate reads that make some genes look more abundant than they really are. Because these duplicates are impossible to tell apart from real biological molecules, they can lead to false positives, especially in low input or low complexity samples. PCR amplification can distort the data, making results look more confident than they actually are. For RNA sequencing methods, library preparation workflow need to be flexible because real samples are rarely perfect. They should work well with low quality or degraded samples like FFPE tissue, should handle a wide range of input amounts, and be compatible with different species, including mouse and human samples. This adaptability ensures that experiments are reproducible, reliable, and able to generate meaningful biological insights across different study designs. Workflow versatility is essential for robust and reproducible RNA sequencing. Proper ribosomal RNA depletion is critical for high-quality RNA sequencing. Workflows that fail to remove rRNA effectively produce low-quality data, often missing rare or low-abundant sequences. They also reduce cost efficiency because more sequencing reads are wasted on rRNA instead of informative transcripts. Finally, poor ribodepletion makes it especially challenging to analyze low input samples, such as those with less than 1 nanogram of total RNA. Effective rRNA depletion is essential for accurate, cost-efficient, and reliable sequencing results. With SmartSeq total RNA library preparation with ZAPR depletion, you get confidence in every RNA sequencing result. The workflow delivers cleaner data by reducing PCR duplicates with optional UMIs for added certainty, especially critical when samples are limited. It's built for real-world challenges, performing reliably with cell-free RNA, FFPE, and a wide input range. And because ZAPR depletion is already included, there are no extra reagents or hidden costs. The result is strand-specific, full-length transcript coverage that captures both coding and non-coding RNA, so you never miss what matters. The SmartSeq workflow is optimized for accurate and reliable library preparation, consisting of 4 key steps. First, reverse transcription converts RNA into cDNA using template switch oligos, providing strand specificity and incorporating UMIs to minimize PCR bias. Next, cDNA amplification generates index libraries which are then depleted of rRNA reads using ZAPR technology, ensuring high-quality informative sequencing data while maintaining the complexity of the samples. Total RNA samples without depletion will mostly have rRNA reads, as can be seen in the figure. Hence, the need to deplete rRNA. With ZAPR depletion, efficient depletion of rRNA is achieved while maintaining the complexity of the samples. SmartSeq library preparation supports a wide range of RNA inputs while maintaining uniform depletion performance across all inputs as shown in figure here. Higher duplicate reads at inputs below 1 nanogram are expected due to high sequencing depth and combined with low RNA input. Strong correlations between technical replicate and across input levels demonstrate excellent reproducibility, underscoring the reliability and minimal bias of the data. We see uniform gene body coverage across the entire input range from 250 picogram to 1 microgram, and it is important as it ensures that reads are evenly distributed across transcripts, allowing accurate quantification of gene expression. It also minimizes 5' or 3' bias, improving the reliability of comparisons across genes, samples, and experimental conditions. SmartSeq library detects not only protein coding gene but other biotypes as well. Capturing this diversity provides a more complete view of gene regulation, cellular state, and disease mechanism beyond protein coding genes alone. Identifying fusion genes in FFPE RNA using RNA sequencing is challenging due to RNA degradation, chemical modification, and fragmentation that reduce read length and complexity. Despite these difficulties, it is critically important because fusion genes are key diagnostic and therapeutic biomarkers, especially in cancer, and RNA sequencing enable their direct detection at the transcript level. We used biologically relevant control material designed to mimic patient samples by containing elk, rat, and ROS1 fusion transcripts in a formalin-fixed, paraffin-embedded format. Detection of fusion biomarkers at low input validate the performance of the assay. To close, today we highlighted what truly matters in total RNA sequencing and how Takara Bio's SmartSeq total RNA technologies addresses these challenges with a versatile and reliable solution. Beyond this, Takara Bio offers a broad NGS portfolio from mRNA and small RNA sequencing to ultra-sensitive immune profiling, along with platform-flexible library prep and high-throughput single-cell solutions like the Shasta single-cell system. Together, we provide the flexibility and performance to support your research at every scale. With this, I will end my talk, and that's good science.
Thanks for joining us for another episode of Teach Me in 10, brought to you by Takara Bio USA. Takara Bio offers a broad portfolio of innovative next-generation sequencing solutions designed to support diverse RNA-Seq applications. For more information and helpful resources, check out the links in the description. And if you enjoyed this episode, don't forget to like and subscribe to catch all of our latest releases.